Alright, enzymes are the topic of our video today, and they're very important because they help reactions in all organisms happen in a timely manner. So nothing that went on in our bodies chemically could happen without enzymes. Now, enzymes are mostly proteins. That's why we're studying it after proteins. One example of a non-protein enzyme is RNA, which is a nucleic acid, but we'll talk about its enzymatic role later. Enzymes are all catalysts, and what that means is that they speed up reactions that already occur naturally. So the reactions that they speed up would normally occur, but much slower. What's useful about enzymes is that they're not used up in a chemical reaction. So theoretically, if you put a catalyst into a reaction, an enzyme, it speeds up the reaction at the end, you could theoretically take out all of the enzyme that you used. And there are thousands of enzymes in every living organism to speed up the reactions that are naturally happening. So what do I mean by speed up a reaction? Well, here's an example. If I have right here two molecules, one molecule that I have over here is glucose, and then the other is fructose, and they're bound together to the disaccharide sucrose. You might be wondering, why doesn't sucrose naturally just break apart via hydrolysis reaction into glucose and fructose? And it does, but it takes a very long time. So with no enzyme, even if we put a bunch of sugar in water and let it sit there for hundreds of years, very little would actually break apart into glucose and fructose. However, if we use the enzyme sucrase, and good thing to notice, any word you might see that ends in ASE is usually an enzyme. ACE is the suffix for enzyme. If you put just a little bit of sucrase in that water, within a very short amount of time, all of the sucrose will be transformed into glucose and fructose. So it takes only a little bit of enzyme to catalyze thousands of reactions because the enzyme is never used up in that chemical reaction. How do enzymes catalyze or speed up reactions? Well, they lower the activation energy. So, for example, if I have a coordinate grid that I drew up, and just for your information, this is going to be a very important diagram for you to remember. You'll see it on the AP test for sure. And energy is on the y-axis. Even for reactions that give off energy, so are exothermic. When you start out with a reactants, maybe here molecule A is bound to molecule B, even in exothermic reactions, you have to put in a little bit of energy to start rearranging these bonds, breaking the bonds to get them to their products. You have to supply an initial amount of energy to get that reaction started. Now, that's usually why heating up chemical reactions will speed up the transition from reactants to products, because the increased kinetic energy of the reactants will allow that reaction to happen. But heating things up in living systems doesn't really work very well, so we need enzymes. And what enzymes do is decrease that initial amount of energy you have to put into the reactants to change them to products. And that initial amount of energy that you have to put into the reactants is called the activation energy. And it's this hump of increasing energy in this diagram. Now, what an enzyme does is it decreases the activation energy, or the energy you have to put into the reactants before they will transition and react and form products. So this red line would represent the energy throughout a reaction without a enzyme, and the yellow line will represent the amount of energy at any time in a reaction with an enzyme. You can see how the activation energy is much less with an enzyme. So let's zoom in and see what enzymatic activity looks like on a small scale. Alright. So, how enzymes work. If this kind of red thing is an enzyme, remember it's a protein. This is its tertiary structure, so 3D, its shape. Remember, and shape determines function in proteins. Every enzyme has a specific substrate or a molecule or a set of molecules that it helps undergo a reaction. So every enzyme will only have, and usually only have, one substrate or one molecule or atom or reactant that it will catalyze. It cannot be used for just anything. It can be used for one specific molecule. For example, sucrase only catalyzes sucrose hydrolysis. It doesn't work on anything else. So the substance this enzyme catalyzes is called the substrate. And this enzyme has a little location where the substrate fits, and that is called the active site. The substrate fits into an enzyme's active site. So in this area, that's why we were talking about R group interactions last time with proteins. The shape of this active site needs to be maintained, otherwise a substrate can't fit into it. That's why the interactions between the R groups that are close to this active site are very important. Because if through one mutation we change one of those amino acids, its R group might change the whole shape of this active site, and it can't work. So we have a substrate, and it comes in and it touches the active site, and when it gets close in proximity to the active site, enzymes go through something called induced fit, which means that once it quote-unquote feels the substrate starting to bind into its active site, it changes shape just slightly to make a better fit, to hold on to the substrate. You can think of that like when you're going to shake hands and somebody's hand touches yours, you kind of squeeze it before you shake their hand to make an even better fit. And once the substrate is fit into its active site, the enzyme catalyzes the reaction and releases the products. Right. So how exactly does that reduce activation energy? Well, usually in one of four ways. One, if there's more than one substrate coming in, or reactant that comes in and binds to an enzyme in a chemical reaction, this enzyme helps to orient those two reactants in such a way where the chemical reaction can happen. Secondly, an enzyme might be stretching the bonds of the reactants that bind to its active site towards the transition state. Third, it provides a better environment for the reaction. So for example, maybe the active site of an enzyme has very many R groups that are acidic, and the chemical reaction that it catalyzes works better in acidic conditions. So that active site of the enzyme provides the right environment. And last, sometimes the enzyme active site will directly participate in a reaction, so which means the R groups of the amino acids that make up the active site might ionically or hydrogen bond to the reactant and help it transition into products. Alright. So that's kind of basic of how enzymes work, but how are they regulated? Well, temperature is very important in regulation of enzyme activity. Generally, if we increase the temperature a little bit, substrates will move around faster and will then fit into the active site quicker and will increase the catalysis. But if we increase the temperature past that point and it gets too hot, the enzymes will denature. So all enzymes usually have an optimal temperature at which they work the best. Just so too, pH is very specific for an enzyme. An enzyme will have a specific pH that it will work best at. If it's too basic or too acidic, it can be denatured as well. In addition, cofactors also help facilitate enzyme activity. So cofactors are non-enzymes that bind to and help an enzyme. And organic cofactors, so cofactors that are organic, made out of carbon, are called coenzymes. So for example, here might be an enzyme, and for it to be able to catalyze the reaction of this substrate into two products, a coenzyme has to fit into the active site along with the substrate to help things along. In addition, some other cofactors might bind to a separate part of the enzyme, and the substrate can't be converted from reactant to product until it binds to the active site of the enzyme and the cofactor binds to the separate site of the enzyme. In addition, there's things called inhibitors. Now, they do just what they say you might think. They reduce an enzyme's activity. So, for example, some types of inhibitors, called competitive inhibitors, are floating around and fit into the active site of an enzyme, just like a substrate would, except the inhibitor just sits there and it blocks that active site from the substrate. So the substrate can't go into the active site and be catalyzed. So they're competing for that space with the substrate. Now, how would you undo the effect of a competitive inhibitor? Well, increase the substrate concentration. The more substrate there is in solution, they're going to begin to outnumber these inhibitors, and more of them will be catalyzed, and the inhibitor's effect will decrease. In other cases, non-competitive inhibitors, kind of like cofactors, bind to a separate part of the enzyme that isn't the active site, but them binding to that place changes the shape of the active site of the enzyme, and then the substrate can't fit in there. So competitive and non-competitive inhibition are two ways that an enzyme's activity is reduced. And lastly, there's allosteric regulators. So I borrowed these pictures because I couldn't draw as well. But one type of allosteric inhibitor, or allosteric regulator, is allosteric inhibitors. Now, what allosteric regulation means is that there are some molecules in cells that kind of behave like non-competitive inhibitors that either inhibit or activate an enzyme. So, for example, certain enzymes have multiple subunits, so they can have 4 different active sites, and they're made of kind of a complex of 4 different enzymatic subunits. Now, chemicals called activators might allosterically activate an enzyme by binding to a separate part of that enzyme complex and changing its shape so that substrate can bind to it. On the other hand, there might be another chemical called an allosteric inhibitor that could bind to the same enzyme complex, but in such a way that its shape is changed so substrate cannot bind to the active site. So those are allosteric activators and inhibitors. Kind of in addition, there's a phenomenon called cooperativity, right, where in the same type of enzyme that has 4 different subunits that can bind 4 different substrates, when one substrate binds to one active site, it activates the 3 other active sites. So once one substrate binds to this enzyme in one subunit, it increases the ability for 3 more substrate molecules to bind to this enzyme and be catalyzed. And lastly, there is what's called enzymatic feedback regulation. So say we have substrate number 1, and we want to create a specific product. Well, a lot of times, to get to that product, a substrate will have to go through several different enzymatic steps. So first, that substrate binds the active site of this blue enzyme, and it catalyzes a reaction in which this red substrate is changed to this green substrate, and the green substrate then has to be catalyzed via a second enzyme from the green substrate to the purple product, and then that purple product is catalyzed by the orange enzyme to an orange product. Well, in feedback control, once we get too much of this purple product, somehow this blue enzyme needs to stop creating green enzyme needs to shut down this whole pathway. So this purple product can actually serve as a non-competitive inhibitor back here for this first enzyme, thus shutting off this enzymatic pathway, creating this orange product. So once enough red is converted to green and green is converted to purple, and purple starts building up, this organism's enzyme might be, hey, we don't need to make any more purple, so that purple will start to competitively inhibit this blue enzyme. And the more purple that there is, the more of these enzymes that it can inhibit. So it depends on concentration of purple. So that kind of in a nutshell is the need to know info about enzymes. If you have any questions, please look over this video. Otherwise, you can ask me or email me at any point. Thanks.